Good afternoon, everybody. I just need to point out that I'm not Rick Beale, but I am chairing. Rick's had a family emergency, so my main job is as timekeeper and fat controller, I guess. But I'll give you straight over to Professor Joe Peters to kick us off.
Well, thank you, Nigel. Well, good afternoon, everyone. Well, it's my pleasure, on behalf of the London and Home Counties Boxer Club, to welcome you all today to this seminar on juvenile kidney disease and to introduce to you the speakers. Now, we're very lucky this afternoon that we have three very eminent speakers, all of whom are noted experts in their respective fields. Our first speaker is Professor Hattie Syme, who is Professor of Veterinary Internal Medicine at the Royal Vet College. She has a special interest in the kidney and has won awards for her work on kidney disease. And she's going to talk to us about veterinary aspects of the disease. Then Dr. Bruce Cattanach will talk. Of course, he's very well known to you. He spent almost all his life in boxers. But he has also, in his professional life, he was director of the Medical Research Council's Mammalian Genetics Unit and has an outstanding record in genetics. So there's nobody better qualified to talk about the genetics of juvenile kidney disease. Then Professor Bill Amos will speak on the hunt for the gene. Now, Bill is Professor of Evolutionary Genetics in the University of Cambridge. He has many scientific interests. He's got very original ideas and he's fizzing with ideas, and we're very lucky that he took an interest in JKD and can give us his perspective. So and then following the talks we're going to have a break for tea, and then there will be a discussion session. So without further ado, hand over to Nigel now.
That's the other one. That's the one.
Yeah. Have you put your mic on, Hattie?
Yes.
Good.
Okay, can everybody hear me?
Yes.
Excellent. So my name's Hattie. I'm going to talk about some of the medical aspects of kidney disease. I've shamelessly stolen pictures of boxers from the internet, so if one of yours comes up, I'm sorry, because I don't have a boxer myself. And also what I'm going to try and do is outline some of the ways that kidney disease may come about and try and explain a little bit about what the kidney does. I'm going to do this at a very basic level. So if you have a medical background, if you have understanding of kidney disease, excuse me if I'm patronising to you, that's not my intention. I'm just trying to explain things at a level that hopefully everybody can understand. So what I want to do is just briefly to talk about what the kidneys do and what happens when they fail. Then to mention a little bit about how we tend to test for kidney disease, and then outline what evidence that we've got that there's a problem in boxers, and then talk about whether this is one disease or many different types of disease. Okay, so what do the kidneys do? Well, the kidneys, as you probably all know, they remove all the waste products from the blood. So all the time, the guts are absorbing all sorts of nutrition, but also sorts of junk as well that the body then needs to get rid of. The products of metabolism, all sorts of waste products are being produced all the time, and the body needs a way of eliminating those. And although the liver does that in some part, the kidney is the main route of excretion from the body. The kidneys are also responsible for controlling the levels of fluid and electrolyte, or the different types of salts within the blood. And because of this role, they also have a function in controlling blood pressure. They produce various different types of hormones that control some of the mineral levels, so calcium and phosphate, but also hormones that control production of red blood cells. So if a patient has kidney disease, they often develop a mild anemia as a result, and of course lots more. So how does this happen? How does this come about? So this is a diagram of a dog's kidney, and ultimately the kidneys produce urine, and the urine goes from the kidney down the ureter to the bladder, where it's then held before elimination. And the kidneys have a massive blood supply. So you've got this arterial supply, and the kidneys receive about 20 to 25% of the blood that's pumped out of the heart, which is pretty amazing when actually the kidneys only weigh about 0.5% of the patient. So they get something like 30 times as much blood as muscle does. And they need that to be able to perform their sort of filtration function. Now, if you look at the kidneys and you look in more detail at this section, what you have is a little duct, which is where the urine comes out and into the renal pelvis. And this duct has numerous nephrons leading into it. Now, nephrons are what we're going to talk about next. Nephrons are the individual functioning units of the kidney. So this is the same diagram at greater magnification. The nephrons, of which there are several feeding into each of these ducts, and each boxer dog would be expected to have 300, 400,000 nephrons in each kidney. So there are lots and lots of these individual units. They have a complex arrangement of blood vessels overlying it. And what I'm going to do now is nephron structure and function is really complex. So I am going to break it down to a very superficial level. And instead of talking about all the individual components of the nephron, we're just going to say, okay, eventually the nephrons feed into the collecting duct, and you have multiple nephrons feeding into each duct. But at its most basic level, a nephron consists of the glomerulus, which is the filter, and the tubules. And we're going to look at what each of these does in turn. I'm not quite sure where I'm supposed to be pointing this at. Okay, so the glomerulus, this is in effect a sieve. And the way that the kidney works is basically the blood gets sieved, and small molecules can pass freely through that sieve. But blood cells and large proteins and things don't get through the holes in the sieve and remain within the bloodstream. Then the fluid and the small molecules that have passed through the sieve get reabsorbed by the tubules. And then eventually urine is produced. So this sieving process, so it's basically on the basis of size. There's a little bit of charge comes into it as well, but cells which are large and proteins which are moderately sized get retained and don't get through this filter, but you've got an awful lot of water and a lot of small molecules that pass through this barrier. And the amount of this filtrate that's formed is massive. So if you think that actually, I said 20-25% of the blood pumped by the heart actually goes to the kidneys, and then about 20% of that actually passes through the filter. There is a limit, because if too much passed through, basically you'd have treacle left in your blood vessels. So about 20% of it passes through. And that means that a human filters about 150 litres a day, or your average boxer about 100 litres per day. So if you think of an oil drum, those hold about 40 gallons, so it's or 200 litres, so it's about half of an oil drum of fluid. But almost all of this then gets reabsorbed. and it gets reabsorbed in the tubules. So you've got your glomerulus, your blood vessel, that allows the small molecules through into the tubule. And then if we look at the tubule in a magnified view, then what the body does is selectively chooses what it wants to take back up. So things like glucose, sugars and things which are going to be useful for the body, those get reabsorbed from the filtrate and get taken back up into the blood. Now it sort of seems like this is kind of an inefficient system. So you throw everything out and then you reabsorb, at least for the fluids, you reabsorb 99% of what you've thrown out. But the advantage of this system is it means that, you know, there might be thousands of different molecules that the body is producing all the time. You can't have a specific way of getting rid of everything that you don't need. It's actually much easier to throw everything out and then take back what the body needs. And that's the process by which the kidney works. Does that make sense? Okay. So, and then the final part is in the collecting duct. Well, a little bit of this happens in the tubules as well. A little bit of fluid gets reabsorbed through the tubules, but then in the collecting duct, lots and lots of fluid gets absorbed. So although lots of fluid gets filtered, 99% of it gets reabsorbed, and so the final volume of urine is relatively small compared to what was filtered. So if we think about what happens when the kidney fails, so now we've got all these individual nephrons, and actually, you know, the dog might have 500,000 of them, but I've just represented a few on this diagram. Basically, what happens in kidney disease, no matter what kidney disease, eventually when the kidney is failing, however the process started, you have loss of lots of those individual nephrons. So you've just got a few functioning nephrons left. And usually we say that you have to lose about 3/4 of the nephrons before you can detect that with conventional blood tests. So unfortunately there's an awful lot of function that's lost before you can detect it in the blood. And people, especially people who are trying to sell you lab tests, tend to say, oh, well, that's because this lab test isn't very good or that lab test isn't very good. But actually, part of the reason that it's difficult to detect that you've got a kidney problem is actually up until a certain point, the nephrons that are remaining, the ones that are still working, actually become supernephrons. So they get a little bit bigger, they work a little bit harder, and they filter a little bit more. So there's some compensation for the fact that other nephrons have died. So as filtration of the blood reduces because you've got loss of nephrons, then basically things that would normally be eliminated in the body accumulate in the blood. So they are present at higher concentrations. And some of these are the things that are toxic and will make a patient with chronic kidney disease feel unwell when they accumulate to high levels. And some of them are used as markers for this patient has kidney disease. So the ones that you are likely to have heard about are creatinine, which is the one that is probably the one that we use most often. Urea, which is sometimes also called BUN, that's a sort of Americanism for it. And then there's a newer test called SDMA, which sometimes people say, oh, SDMA is a better test, and blah, blah. They're basically measures of the same thing. They are measures of this process of filtration, and when the filtration process fails, these substances accumulate in the blood. So although there may be small differences between them, they're essentially measuring the same function, and none of them are going to have great advantages one over the other. And often the differences are basically if you want to make a test more sensitive, you just make your cutoff lower. So then you're going to get more dogs that test abnormal, but they may or may not have the disease. So you may have an increase in sensitivity of your test, but with a reduction in specificity. So the remaining nephrons do compensate a little bit. They get bigger. The glomerulus actually gets stretched, and it also functions at higher pressure. And if you've got higher pressure within these blood vessels, a little bit more protein will get squeezed through the filter, and so you may get a little bit more protein in your urine. And the other thing that happens is, as the kidneys start to fail, you get more dilute urine produced. And that seems very counterintuitive. So if the kidneys are failing, why do you produce more urine, not less urine? But the reason for that is because even though less is being filtered, remember, usually 99% of what's filtered is reabsorbed by the kidney. And when you've only got a few remaining nephrons, they're less effective at reabsorbing stuff. And so now, instead of being able to reabsorb 99% of what was filtered, if they can only reabsorb 97% or 95%, then actually 4 times as much urine ends up being produced. So that's why there's production of more but more dilute urine. So what evidence do we have of kidney disease in boxers? Well, there have always been a sort of sporadic case reports. So case reports are things that are written up in the literature, in the scientific literature, where somebody describes an individual case or a small number of cases that are well characterized. So somebody's done a lot of testing on them, they've sent samples to the pathologist, and they've identified something that hasn't been described before. So there had been sporadic descriptions of boxers with kidney disease, and then somebody called Marge Chandler, when she was up at University of Edinburgh, contacted me, contacted people at the other vet schools, and said, I'd like to put a case series together, can you send me the details of any boxers you've seen in the last 5 years with kidney disease? And we somewhat arbitrarily decided that we would include boxers that were less than 5 years, because boxers, just like every other breed, there is some kidney disease that happens with aging and just by the by. I mean, there are lots of different types of kidney disease. So what we were trying to do was to skew it towards younger animals, which were more likely to have a congenital or a familial form of the disease. And so included in the group we had boxers from 4 months to 5 years. So things that were, I think, interesting were that there was a preponderance of females in the series that we put together, and that quite a few of these dogs presented, they had a history of incontinence or of urinary tract infection, and those as well as kidney disease. And this is one of the things where it's a real chicken and egg, because patients with incontinence are more likely to get urinary tract infections. If they don't have a very good sphincter to retain urine in their bladder appropriately, and urine can in effect leak out, then at times it's probably also leaking in the opposite direction or taking urine from near the outside, near the vulva, which is not a sterile place, and washing bacteria into the bladder with it. So patients with incontinence are predisposed to urinary tract infections, but also we will see some dogs that maybe they don't have the strongest sphincter in the world, but they're fine until they get an infection, and then they'll present with incontinence. And in addition, urinary tract infections, although most urinary tract infections, when they're located in the bladder, they don't cause a problem to the kidney. They can go up the ureters into the kidney itself and cause an infection there, and they can actually be a cause of kidney disease. But kidneys that are already diseased are more vulnerable to this actually happening. So everything sort of can occur in different orders. And actually, so the ultrasound changes, so most of the dogs were ultrasounded, examined by that. They had lots of changes, but they were very non-specific. So a diseased kidney doesn't have the beautiful sort of kidney architecture that we would expect, but it doesn't tell you what made that kidney diseased. It just tells you that disease is there. And there was pathology performed in only about 9 dogs, or only 9 dogs. It was very non-specific, the results. And certainly when renal diseases advance, when kidney diseases advance, what you end up seeing, especially if you do a sort of needle biopsy, so a very small sample, you end up with scar tissue that says, okay, this is a diseased kidney, but it doesn't necessarily tell you what started the whole process. Okay, so another paper that has been published is a study from Sweden, and in Sweden there is a requirement to have your dog insured. So actually something like 70% of the population are insured.
Well, I'm so sorry to interrupt you. I'm really struggling to hear you because it's so noisy next door, and I wondered if somebody could just nip in and just ask them if they can, because it's just really difficult to hear you properly.
Does that go up?
Yeah, or I can try and speak up louder. I didn't realise I was speaking quietly. Right. And is anybody else having a difficulty? If you're not,
There's 3 seats down here, 4 seats down here.
Some at the front then.
but also if you're losing me, just wave and I'll try and speak up.
Sorry.
So the other study that has been published was looking at dogs in Sweden. So in Sweden, it's required that your dog has insurance at least up until the age of 10, and a lot of people are insured with just one major insurance company, which means that they've been able to look at insurance records and get lots of information about what diseases different breeds of dogs get, and what diseases they have and what diseases they eventually die of. And looking at kidney disease in the Swedish boxer population, or in Swedish dogs overall, boxers came out as number 3 in terms of having kidney disease, and number 4 in terms of dogs actually dying of their kidney disease. Now, the limitation is that these dogs were seen by vets in general practice, and so the vet wrote on the record that the dog had kidney disease. We have no idea of how detailed that diagnosis was, whether that was more their suspicion or if they'd proven it with blood tests or anything like that. But nonetheless, the boxer did feature in that. And what they found was that if you assume that the dogs had a 10-year lifespan, then 1.6% of the dog population as a whole develop kidney disease over 10 years, and 3.6% of the boxers. So sort of more than twice that of the general population. Still not every boxer gets kidney disease, mercifully, but more than the general population, and 2.3% of them died of the disease. Now, as a comparator, I'm sure that you're very well versed on heart disease in boxers and things like that. Looking at the same database of dogs in Sweden, then the prevalence of heart disease was 4.6%. So about twice what they had of kidney disease. But I know that you all know that heart disease is, an important problem, and in spite of that, because these numbers can look quite small, that's still 4.6% of the dogs, and it's 2.3% that have kidney disease. Again, and this is a recurring theme, it seems to be more common in the females than the males. And the average age of death due to kidney disease in the boxers was 5 years. But there was a big spread. So what are some of the specific causes that have been described of kidney disease in boxers? Frustratingly, so there are very few that have had very good pathological examination. So a complete pathological examination would be when a patient's died and you have a pathologist that both examines the body but also takes the kidney and looks at it under the microscope and tries to determine what the characteristics of the underlying disease were. One of the terms that is described is something called renal dysplasia. Now, renal dysplasia, when an animal is born, the kidneys aren't actually fully developed. In the first few weeks of life, the kidneys continue to develop. And the kidneys, those structures that we were talking about, the glomerulus and the tubules, look different. They look immature in a fetus compared to a patient that is adult. And the characteristic of renal dysplasia is that you still have these fetal or sort of baby glomerulus and nephrons in an adult animal that where they should have matured. The problem about renal dysplasia, the term renal dysplasia, is a lot of people, and even at a relatively specialist level in the veterinary community, don't really understand this term and don't really understand what it means. So at its best, this is a very specific entity, but it tends to get bandied around, and especially imagers, so ultrasonographers and things, think this term means a young dog with bad-looking kidneys. And so a lot of people say, oh yeah, it's got dysplasia. And I'm like, okay, and what does dysplasia look like under the microscope? Or they have no idea. It's just a term that's applied. And that makes it unfortunate in terms of actually trying to understand what they really have. And with the ultrasound, you can really only say, this is a pretty knackered-looking kidney that doesn't have the beautiful structures that it should have. You cannot make a microscopic diagnosis with your ultrasound. The other thing that has been described, and actually this was a better characterised paper, again a group in Scandinavia described this, and they described dogs as having what they call the Ask-Upmar kidney or reflux nephropathy. And what this is, and this is a diagram of a human anatomy. So humans have a slightly different arrangement of in their renal pelvis. But in a normal human, when your bladder fills, then actually that acts as a sort of stop valve to stop urine going back up the ureter into the kidney. And what happens in this disease is that this valve is somehow defective, and that defective valve doesn't cause a problem in the kidneys in itself, but what it means is that if this patient has a urinary tract infection, then infection is more likely to course up the ureter into the kidney, and you get a sort of characteristic pattern of scarring within the kidney, which is very suggestive for this. But it's difficult because this is actually diagnosed in people by getting you to pee on video camera and to actually show with a radio marker and to actually show that the urine's doing this, which has never been done in dogs. But some of the dogs do have this sort of characteristic pattern of scarring. Okay, how am I doing for time? Are you about to? Okay, should be okay. As long as you haven't got too many questions, but we've got lots of time for questions at the end, I understand. Okay, so one of the things that comes up is to think about when we're talking about renal disease in boxers or kidney disease in boxers, is this one disease or many disease? If you think of a different example, and you've got a lame boxer, like this one stolen from the internet, it's easy to appreciate that this lame boxer might be carrying its leg. There might be all sorts of different problems. It might have dislocated its hip, it might have a nail in its paw, it could have a fracture, it probably doesn't have a fracture, hopefully it hasn't got a bone tumour, or it could have ruptured its cruciate, so the ligament in its knee. That would be my favoured hypothesis for this boxer. Now, we think that cruciate disease, there's probably a genetic component to that because there are certain breeds that do it, and because dogs that do it quite often do both of their knees, one after the other. So there's probably a genetic component to it. But if you tried to find the gene for cruciate disease and you just took all lame dogs, you'd really struggle because you've got an assortment of everything in there. You've got to be pretty comfortable that you're looking at dogs that have just got cruciate disease before you start searching for the gene. So if we think about kidney disease and we've got our sick boxer with failing kidneys, then we essentially have the same problem, because we can look at the dog and we can say, okay, these kidneys are failing, but they could be failing for a number of different reasons. So there are a few things that we might see. So we've got pyelonephritis. Pyelonephritis is a posh word that means you've got an infection that has spread from the bladder up into the kidney. Pyelonephritis is a difficult one because that can happen spontaneously, or it can happen just bad luck because you had a bladder infection and it rose up, or like I was showing with the abnormal valves, you could have a patient that was genetically predisposed and was more likely to get pyelonephritis as a result of that faulty anatomy. You can get glomerulonephritis. Now, glomerulonephritis is a disease, an immune-mediated disease, where the body attacks itself, where the patient develops a disease. Sometimes it's triggered by something else, sometimes it's triggered by an infection, and sometimes we can't find any underlying trigger and it seems to be idiopathic. Now, generally, this presents in a slightly different way because the immune complexes, because they're particularly in the glomerulus, that's the sieve I was telling you about. Basically, the holes in the sieve get much bigger and lots of protein leaks out into the urine. So usually we get an indication that it's more likely to be an immune-mediated disease because the patient has massive amounts of protein in the urine. we get old dog chronic kidney disease, just like we get old human kidney disease. That's certainly going to happen. And then you get things that typically present in a more acute manner. So in a patient that was fine one day, really ill the next, so you get things like toxins. I've just put up as an example, but dogs can get acute kidney injury from eating grapes or raisins, or from drugs. And this is one of the things where you can have problems when vets make diagnosis of kidney disease on the basis of blood tests. They can be mistaken sometimes if, say, the patient's taking lots of diuretics or water tablets, because if the patient's very dehydrated, that can make the kidney values go up. And so there can be misdiagnosis of kidney disease on that basis. And then, obviously, there are some genetic causes of kidney disease. Now, the example I've put here, polycystic kidney disease, that's one that is definitely not what the boxers have. I've put this as an example because this is what Persian cats have, and the advantage of this disease is that it's really, really characteristic. You can do an ultrasound and you can see the fluid-filled cyst in the kidney, and you can say, that's what the patient has. So with the cats, with the Persians, even before they developed a genetic test, they were able to do ultrasound screening, identify the patients that were affected, and not use them in their breeding programs. But certainly genetics. So some genetic tests are genetic diseases are very characteristic, and others may not be. So this is Hattie's wish list for what we need going forward. I think we do need better characterization of the problems that we're dealing with, and to ensure that it's only a single disease. Now, I mean, boxers as a whole, there will always be dogs that are there are all sorts of different kidney diseases. I'm pretty confident that there is a genetic boxer kidney disease. There might be more than one. But in terms of trying to find that gene, it's obviously going to be easier if you've got a purer population where all the patients that are affected have the genetic form of the disease, and you've weeded out everything that's got kidney disease for some other reason. And ideally, to do that, you'd have specialist histopathology. Well, I guess that's not ideal because the dog is either dead or having a very expensive diagnostic test. But in terms of characterising the disease, that would be a step forward, and I'm going to tell you something about that in a moment. And although I say that we need this, and this is the conventional or the better trodden path in terms of identifying disease, there certainly are situations where a disease has been identified, maybe not that well characterized, but they've been able to find the gene and have then also worked in the opposite direction and said, okay, this gene, we know it causes a problem in these patients. Now there are other patients that also have this gene. Let's start looking at them, and lo and behold, actually some of those animals also have a disease, but it's mild enough that it wouldn't otherwise have been recognised. So it is possible to work in both directions. In terms of better characterisation of the kidney disease, there is somebody at the Ohio State University that's got a grant from the American Kennel Club to study the disease in boxers. And what she is able to provide is analysis of the renal biopsy. She's not going to pay for the biopsy to be collected, but in terms of the analysis, actually renal histopathology, which includes doing an electron microscopy, is hugely expensive. So that's a test that would cost $850 to be done, and it will be done free of charge if people can get samples. I thought it was only going to be open to AKC registered dogs, but I emailed her prior to coming here, and she will, any dog with a pedigree is acceptable in terms of that study. And just in case you thought it was all doom and gloom and everything was really bad, I've got a colleague that I work with in genetics, and she's looking at the boxer dogs, but she's using boxers as her control population because they don't get diabetes. So I just thought I'd end up in some good news for you. And boxers are very underrepresented in the diabetes study. So if anybody's interested, this is the link to, they've got a website, they've got a Facebook page, and they're studying diabetes, but they're the boxers are the good guys. So I'd obviously not a boxer. This is my own dog who had the temerity to die of kidney disease, although he was 18 and a half at the time.
We prefer to take questions after the three speakers have spoken, particularly as some of your questions might be covered by what's coming. So try and hold those in your head. But thank you, Hattie.
Yeah.
Yes, I'm warm. I was trying to work out how to do it. Oh, yeah. Bruce. Okay, Dr. Bruce Cattanach.
I brought my own stick in case anybody misbehaves. Where does one start here? First, I've put my email up, and if anybody wants any of the slides I'm showing, just email me and I'll send them to you. There's quite a lot of slides which I'll flip over because I can't do everything in 30 minutes, but the bigger story is there on the emails, on the PowerPoint slides. Right, now, as you all know, we have this boxer kidney disease in our dogs. It first arose, as we know it today, about 8 years ago. And since then, I think you probably all understand, it's been a bit of a bumpy ride, for me at least. Different views have been put forward at individual level, at breed club level, international breed club level, uncertain as to whether JKD exists, whether it is in boxers only, whether it's inherited, whether it has a recessive gene inheritance, and indeed whether it is frequent enough, and this is the key one, frequent enough to be worth bothering about. So you've got all these opinions, and that, I guess it is my duty to do today to try and present the case that it is certainly worth dealing with. Now, I need a thingy.
Oh, there it was on the corner. There.
Yeah, it doesn't always work.
There's a slight delay. Press it once.
There we go. Yes, now a problem for me is while one would like to do everything truly scientifically with veterinary schools doing steady diagnosis, being able to bring dogs back and get everything standardized, that's not the way I can do anything. I have to rely 100% upon you people providing me with information, and I have to use every scrap of information I can to try and make a story which I can understand genetically and then present on to you. So at that level, I'm going to look back to the first incident of cases of kidney disease of some kind, possible kidney disease, that I met throughout my lifetime. First of all, this is a key one, Leslie Boyle, late Leslie Boyle, Camsail boxers, came to me in the middle of the PA era, the neuropathy period in boxers. She'd helped a lot with that, and she came and said, I'm wondering about a couple of litters I've got where puppies have been dying, dogs have been dying as they got older from a kidney disease. She showed me the pedigrees, and horror of horrors, they were shown close inbreeding on her champion dog of the day. 2 pedigrees, does that make a story? Meh. But I told her that didn't seem a good story, and she withdrew the dog from stud and stopped that section of her breeding. As far as I know, there have not been any further cases from that source anywhere in Britain. Then, over the years, there was news that the Swedes were collecting information on kidney disease in all breeds and publishing the pedigrees or the names of dogs which had produced and declaring them not fit for breeding any further. Boxers were included in that, very much so, and some of them were of British stock. I was able to get these British pedigrees, I looked at them, I couldn't see anything there at all. You know, there were a mishmash of all sorts of breeding. So I didn't take it too seriously, especially as we were diagnosing breed after breed with having kidney disease. But then I was made aware of a group in America, and I got those pedigrees, and I looked at them, and I thought, wow, well, there's maybe something a bit more here, because about half of them, maybe a bit more, showed inbreeding on one particular top male. And that looked quite worrying, but then there were others. Now, were they just spontaneous ones, such as Hattie been talking about, or did it mean there was a gene, but it was all over the place? Couldn't really tell. But anyway, there was a bunch of money collected for research on this, known as the Tukey story, one of the dogs that got this problem, and that all the stuff went to Kirsten Lindblad Toad, the Broad Institute, to study. She didn't get enough, she didn't find the gene which she was looking for. Then in the UK, back here again, I heard about a mother and son getting kidney disease. Could this be the case? I never saw the pedigrees here, but mother and son both getting the kidney disease, living in the same household at the same time. That didn't sound too much to worry about. It could be anything. And then Sheila Cartwright on the Steve Dean Health Committee, last Health Committee, kept on reading. She had heard about kidney disease in boxers throughout much of her life, but she says there were all different diagnoses. She couldn't tell what was going on, but there must be something to look at. So this was at the end of the Steve Dean Committee. I'd had enough, frankly, having dealt with cardiomyopathy for years, and didn't want to do any more about it. Somebody else did. And that was the end of that as far as I was concerned. Yeah. Oh God, we're going back again.
That's right. That's it.
Oh, go forward now.
Should be down.
You've got to
At the
mention
bottom,
Sharon.
Sharon McCurdy, you can't see it.
You've got to
Oh,
mention
sorry.
Sharon
Yeah.
at the bottom.
I
Yeah.
can't see that, yeah.
Sharon Bombshell.
Anyway, following on this line, am I supposed to be able to change this or not?
You can.
It's just the bottom.
Yeah, that's what I've been trying to do.
Press the
I
button.
put it in, it doesn't always work.
Oh, there you go, pointed at. Right, so going on, what is the evidence of a disease in boxers? Well, you've heard quite a lot of this from Hattie The veterinary publications, she mentioned the Askap Nordman, the agrarian insurance in Sweden, again, but then we've got all the cases I'll be talking about abroad, here and abroad, but the real question for me, is it inherited? Now this is the key bit that I am doing, adding to this sort of veterinary stuff, diagnostic stuff that Hattie's doing. I'm looking at it as a geneticist, first and foremost. Is it inherited? And I'm not obviously going to show all the pedigrees. We've got hundreds of pedigrees. I've just picked using some examples to explain the situation. You've got a pedigree of a case. Its parents were marked in blue as producers, and Lynn Kinkler, who is on the Boxer JKD website and the group who has collated all these pedigrees, has put in extra information such as she can. So this dog is the sire of 3 cases. So this backs up that he's produced this one. He's also sire of dogs which have produced JKD. So this is pretty established. And then the key point is that this case is showing inbreeding on this particular dog. This dog back to his granddaughter. More information up there. And this is just one pedigree, but we've got lots and lots of pedigrees. Here's another one. If we say producer A, here, producer O2, here. Oh, I've got 3 now. There we go. A, here, doubling up on dog A. This is the pattern we see in these UK pedigrees, and you can add also in continental pedigrees and American pedigrees. This is what we see. So we can also have it where the dogs appear to be unrelated, but this one is producing, that one's producing, so there's no worry about, you know, this coming through to here. And then one might find that there's a dog further back in the pedigree, common to both these, and that if you see it often enough, you can conclude that dog X is a producer as well. And you can follow this back as the pedigrees get wider and wider, separate family groups merging together, and you get the disease coming out. But always there is inbreeding at some level, except in the situation of where dogs are totally unrelated, as in, say, a German one and a UK one, from a family that is producing JKD, from a family that is producing JKD. You put them together, total outcross it appears, but you still can get the disease. So just going back now to the links to earlier observations, Leslie was case, she stopped it in this country, but some of her dogs had gone abroad at some point, and they formed nests of kidney disease in other countries, crossing to each other and crossing to native dogs. So this also would have been JKD as we know it today, because these are the current ones fit in here. Then the Swedish cases we saw, they have amplified up the number of cases by a large amount, and we find they fit into ours without any difficulty. We've got overlaps of Swedish and British dogs. In fact, one of our early cases going out to Sweden was diagnosed as RD and put into the Swedish group. The US cases, we had a case just a few weeks, months ago, in which the mother actually traced back to the key dog that I'd seen before, and the father was a British dog of JKD lines. So we're getting a mixture of dogs from any origin. Then there's Marge Chandler's cases, which was mentioned. She sent me the pedigrees at some early point, asked if there was any evidence of inheritance, and it was this mixed bag of thing. You couldn't tell anything. But in view of what we have now, they fit in precisely, they interact with our pedigrees, they're all part of the same thing. So Sheila, we don't have any of her dogs at that time, or dogs she knew about at that time, but she did have a point. So just a summation of the year, you see this, most commonly found with inbreeding. You can also get it by doubling up on dogs that are not related, but they have come from JKD families. And one can trace back, really back to the beginning of JKD, in this country at least, to see where it's come from. This is bringing in dogs that have been exported years ago. And you can see they all link up together. And to me, this all indicates or establishes that we have a recessive gene. Now, if one, what's happening now?
Just click on the resume slide.
Yeah. Do you want to? Yeah.
Yeah, what did you do?
I did.
Oh, right. Okay. So that's a summation of the UK pedigrees. Yeah, and all pedigrees. We've got JKD throughout the world, literally. International crosses can produce it. We find RD cases and JKD cases in the same litters. That's in Norwegian stuff. And it all is indicating that we have one inherited kidney disease in boxers with the same gene, despite the various diagnostic criteria that Hattie was just describing. So this is the, I'm going to hammer, we're talking about a recessive gene. Right, now the question is, one expects with a recessive gene 25%, as I'll show you in a minute. But we've got the practical difficulties, such as Hattie has talked about. And of course there is reporting absences too, likely to be occurred. So it makes life very different. Have I missed the slide?
Yeah, it's
Maybe if
Yeah, I
Kirstine
think
clicks through
Kirstine,
them for
yeah,
you,
I think
if you just
All
say
right.
next, because
I'll
that
wait
seems a
for
bit
you.
dodgy.
It's better than having two hands. Right, so that's the conclusion, and the next one, sorry?
There.
Yeah. So just to be sure everybody knows what we're talking about, recessive gene. Here, JKD, we call the gene KD, we have a plus for the normal gene. And this is the dog that is a carrier, if you like, got one normal gene, one kidney gene. If you make the two together, you get this 25%, 50%, 25% of affected dogs, two blues, carriers, one and one, and the normal ones as well. So this is the figure that is important. We expect to get 25% with the kidney disease. Though we have the problem that they can occur at different ages, different diagnoses and everything, so there are problems. Here are the problems. You get range of ages, etc. So there's lots of things that go wrong. But the important bit to me, one important bit, is there a big sex difference. More common in females than males. Now in the UK, it is whacking great, 4-to-1 ratio. I could hardly believe that, so I got some of the Swedish data, and well, it's just about the same. And going to Norway, I don't know the numbers, but it was a bit lower. Now that being lower attracts my attention. It's not a problem, it attracts attention. So going on from there, what more can we get? Right. So I thought I'd look at the dogs of different ages. So we've got females up here, males down here, numbers of animals and the ages. And you see, first of all, it's very obvious, there's your 4:1 ratio, a lot of them. And the next thing is that I asked if uterine tract infections could be responsible. And uterine tract infections are occurring in some females, more or less in proportion to the figure there, but there's only a tiny number relative to here. If you take them away, you're not going to get this figure. So females are still more evident. Here we see again that kidney disease can be diagnosed very early on. Got cases down below 6 months, but most of them are juvenile, and this applies to the males too. Some of the vets in general practice ultrasound scan them as well, and we find a range of kidney abnormalities, missing kidneys, small kidneys, scarred kidneys, even cystic kidneys among these dogs, all of which seem to have the same family group, the same genetic background, hopefully the same gene. And so this is saying that whatever is leading to kidney disease is possibly occurring earlier. And here you can see so much early, right at the very beginning. There is this skew, at least certainly up to 12 months there is a skew, is constant, more females than males. So something else is going on. What is it? Can we follow this further back? So UTI is not involved. The key bit is that tail of the distribution. Tail down here, tail down there. And interesting, the Norwegian data had a lot more males, I'm told, at later ages. So this is the bit I think is the most important of the lot. This is a shortage of males. What is happening? Are males more severely affected so that they die very early on? Or is it that they're less affected and go on and don't produce the disease? There is possibly some evidence to support this from human data, where I'm told that this kind of abnormality is commonly more seen more frequently in males than females. So maybe that's that. But I have no direct evidence at all. What evidence do you expect? You'd expect low litter sizes if they're dying before birth. You'd expect dying puppies. You'd expect fading puppies, things going wrong at this very early age. I don't have any access to that information, barring one litter where one new breeder had zealously screened puppies that she thought there was a problem with. In the litter there was one small runty puppy and one fading puppy, which were post-mortem, and the kidneys were examined and said to be kidney disease, a kidney disease. 2 other puppies, at least in the litter, developed what we call JKD later. So pretty certain that is the case. But there's one puppy was a female, so not a male, so it doesn't help us at all to understanding what is going on here. Now, as to here, I think you could say you have evidence. You've got that tail trailing on to later ages of dogs of the same breeding, being litter mates of others, all having the disease. So I think there are positive indications that this could be occurring. and that is crucially important. The implications, if the males from affected litters are having 2 doses of the gene, so they can go on and survive and breed, then it is spreading the disease around very much more rapidly, and you don't know anything about it. So that is the problem. And then there is the other problem for the people looking for the gene. If you have a population of females with the disease, 2 doses of it, sorry, there's a females, population of dogs with 2 doses of the gene which are affected, and you want to see what is different from them at the DNA level from a normal population of dogs, and the normal population of dogs has got ones with 2 doses as well. You're comparing like with like. So it's making life very much more complicated. Now we're going off again. Oh, sorry. Again. No, you're going to have to sort it again because the always has.
There we go.
There we go. So breeding recommendations, well, some of them I've given out before, but there is the old-fashioned way of avoiding. One, breeding from animals that produced JKD. I mean, that's not mind brain. If you don't want to get light-eyed puppies, you don't use dogs that produce light eyes. If you don't want JKD, you don't use dogs that produce JKD. Very simple. Now I've added this one in too, on the litters where we've got affected ones. That has got more serious now in view of the recent discoveries that they are at higher risk, and it's probably with females as well as males, don't know, but it's definitely males. But not many people are going to use dogs at stud, which come from litters of JKD, so it's a minor point. Then avoiding inbreeding as far as possible. The more closer they are, they're more likely to turn up the disease, JKD, or anything else, in fact, if there's anything else in there. Not doubling up on dogs which both sides produce JKD. We've covered that kind of angle. But the other cautionary thing is not to select too hard. Now with cardiomyopathy, I think people did select too hard. They went out everything, which was a shame. But here, one is just going to have to grit one's teeth and breed on from descendants of producers. Otherwise, as you know, in this breed, we haven't got anywhere else to go. The other couple of cautionary things, which are a little bit independent, is avoiding the use of excess use of males. All the eggs going into one basket. If there's any problem with these dogs other than JKD, it's going to come out in a couple of generations' time. So just be careful about that. Now, as to imports, they've got JKD abroad. We've got it here. It seems to be pretty high frequency over here. Maybe dogs from abroad are not going to make things any worse, but all you can do is caution, try to find out about them so you're not bringing in stuff that has produced it already, as has happened actually. Then just general advice. Urine-specific gravity testing, as Marge Chandler did for us a few years ago, it would be a good idea to check everything you can to try and find out what your state is, if your puppies are all right, if your stock, if sold is all right. And this is a good one. A good proportion of the people reporting JKD cases to me say, we saw there was something wrong from a puppy. It was a run to the litter. I wanted a little one, pet people. Breeders won't be going for that too much, which is maybe why you don't get so many cases reported from breeders. But anyway, so I think that is something to avoid at all costs for any kind of animal breeding. Recording fading puppies. I'm looking to see where these missing males are, where they've gone. Are they really being lost here, or are they occurring later? So look for missing puppies, record the litter sizes, and this is as much for analysis, genetic analysis, as your own point of view, but it's a guideline to where things may not be right. Now, again, this is then following up in later ages, and all of this actually should be reported. But for research purposes, I don't think it's a matter. Kirsty? Oh, go on. I'm doing it without even looking. So, what I think is needed research, I think the needed stuff is to find out why we have a shortage of males. Are they fading puppies, dying puppies early on, which doesn't make much difference to the breed. It's a horrible waste of puppies. But if are they dying later, and then the only way we can check after that is to look at the kidneys of dogs which are dying at old age. All dogs, it's the normal ones we're looking at. And then the ultimate one is to find the gene. Now, I think that is basically my story, and it's really up to you guys to decide what you want to do. I've told you the story as best as I can, the most information I can get out of it, and you having to decide whether you're going to live with a breed which produces puppies which are dying like this, and the next pretty ghastly, horrible death as they slowly poison themselves to death. So that is the story. It's really up to you. My bit is over. Just acknowledgements to the Boxer JKG group, which is international group, America, Sweden, and here, who have collated all the pedigrees and got all the stuff with me. Then, of course, all the owners in here and abroad who have contributed material. Leslie Boyle, I think, needs attention because she was the first to really show that there was a problem. Sharon McCurdy, whose sad experiences brought JKD to light. She had a whole bunches of stuff in the beginning, helped find lots of early cases. And then I should mention Kirsty, who's done a lot of work on these slides and helping me with this stuff. And last but not least, my geneticist colleague and wife, who has kept everything together over this past quite difficult 8 years. Thank you.
Thank you.
Yes, please, off. Yes, off, please.
You aren't directly under it.
I know I am.
It's moving.
On these trigger device, thank you. Right. Okay, forward or just back, Jan?
Down is forward. The down arrow. I
that
think
one.
if you press that one, it takes it off completely. So it's down is forward.
Yeah, okay.
Yeah.
So, over to Bill Amos now, who's not going to use the sound system, his voice and Thank you, Bill.
I think so, yeah. Okay.
Thank you.
So I'm not sure that you really need lots of technical genetics at this point in a hot room, so I'll try to keep this to an extremely noddy level if possible. This is really sharing my experiences after severe arm injuries from the amount of twisting that Bruce exerted. I met Bruce on the Royal Society panel, which was looking at inbreeding in pedigree dogs, and afterwards he plagued me with innumerable emails saying, why don't you look at this, why don't you look at this? And I said, I've never worked on dogs in my life. And he said, no, come on, you've got to look at this. And in the end, I reneged and started what I thought was going to be a small study, but it seems to expand. So just some really basics of how you might go about looking for the gene for a nasty disease. This would be a classic pattern that you try to look for. You get a bunch of diseased animals, each of which carry 2 copies of a particular gene. And here's a bunch of healthy controls. And from the colour coding, I hope you can see by eye that the diseased ones have a lot more red compared with the healthy. And you might go away and say, oh, crikey, we found a nice association. And that all works well in very simple circumstances. But I think most of what we've heard before is that the JKD scenario isn't anything but simple, what with the skewed sex ratio, etc. And in fact, I think well, I'm going to tell you the story of where I've got to and how I've been looking. So it's not as simple in pedigree dogs, if nothing else, because of the extremely strange pedigrees that emerge. They're nothing like natural populations that I'm used to working with. Obviously, you get the recycling of top studs who come into pedigrees again and again. So, for example, if we consider, this is my diagram of what's meant to be the pedigree of two different breeders, each of whom has got a top stud dog who they've used quite a bit. And as you can see immediately, that if the top stud dog here is a red and yellow, that not every gene in this pedigree is going to be red or yellow, but there are more reds and yellows in the animals produced by this breeder compared with this one over here, who has quite a few blues and yellows filtering through. Now, if you just look at that pattern, you'd say, if you just treat these as random samples, you'd say, these are completely different from these genetically. And it's just because they're much more related in samples from a particular breeder. And this means, for example, I've tried to be anonymous in some cases. Maybe you'll recognise anyway. If we've got a number of different breeders here, and they've each got their studs, and they've each got their pedigrees, maybe some of them are more reluctant to report than others, so maybe some of them don't report cases of JKD, whereas others do. So here's a perhaps worst case scenario where we've got 4 breeders, and only one of them reports cases of JKD. This unfortunately means that you open up to environmental associations. So here perhaps these breeders all feed their dogs on different breeds of dog food, different types of dog food, and so here we see that breeder 3 is producing quite a few closely related dogs, so they're genetically similar, and we've got lots of JKD cases, but they've all eaten the same dog food, so you might immediately think, oh, perhaps that dog food's a problem. Equally, I've had emails from somebody who's convinced that there is a vaccine that is used, I can't remember the name of it off the top of my head, but it's suggesting that, okay, yes, okay, so a particular vaccine, and says that there's an association there. And the trouble is, statistically, many of these things are really difficult to disentangle. Many of you may remember all the stuff with MMR vaccine. It's really difficult because there you have a vaccine that is given to human children at exactly the same time as autism is diagnosed. And people, oh crikey, look, I gave my child the vaccine and it was diagnosed with autism, and they assume that there's a connection. And you can get a lot of connections which are apparent, but are actually due more to chance or spurious associations due to the structure of the data. So there's a warning there. So there are a number of possibilities for what can be the cause of JKD, and there's a whole series. Bruce has really emphasised the idea that there's one gene and that it's recessive, but there are other factors that could come in. There's inbreeding depression. We all know that there's a possible risk, particularly in pedigree dogs. There are environmental factors. It may be that you have to have a particular gene, but you only develop the disease if you get an injection, or if you're given a particular diet, or if you're stressed at a particular time in your life. It might be that there's one gene, or it might be that there's one gene plus the environment. There might be 2 or more genes. I float the possibility, it's slightly poo-pooed by the other speakers, I haven't thought about it much, and I'm sure they're right, but the possibility that there's a sex linkage effect, that there's a gene on one of the sex chromosomes that it sort of provides the right background on which another gene can cause the disease. And we can add more and more and more things, and really important is what Bruce was talking about, which is that there are potentially multiple causes. And it's really difficult to track down a gene when you've got half the cases have one gene and the other half have a different gene or an environmental effect, because that just muddies the waters and makes it a really fuzzy picture that's difficult to resolve. So it's really difficult if there are multiple genes or if there are environmental effects, unless you can control for this. In order to disentangle this, you really need lots and lots of samples, so that's what I've been trying to collect. And Bruce has given me a whole load from previous JKD studies. We've also had a lot of requests going out, and many of you, I think, have contributed swabs from dogs, both unaffected and affected. A recent Facebook plea generated a whole lot more. It's particularly interesting to have affected and unaffected siblings, because in general that can control quite a lot for the environment. They will have grown up under the same circumstances, they'll probably have been vaccinated in the same way, they'll have been given the same food. So if one of them gets the disease and the other one doesn't, it's much more likely to be genetic. So my sample set has an original study on tracking down brindle, the original JKD study, lots of swabs, and the Facebook plea, and this has resulted in about 100 cases and 1,100 controls. The controls are really important. You might say, why on earth are you bothering with that number of controls? But actually, the more you understand what healthy dogs look like, the easier it is to find a subtle difference that is associated with the disease. Really important is that we've got samples from all over the place. We've got lots of samples, obviously from GB, but we've also got American, French, Scandinavian, Australian, and New Zealand samples. And this is really important because the dogs are so related to each other that actually unpicking what's due to the fact they're just related rather than that the particular gene is being shared is quite difficult. So it's really nice to have as far as unrelated dogs as possible. It helps you unpick what's truly genetic and what's just simply they all come from the same breeder. So diverse lineages, large numbers, sibling pairs, these are all things that can help us try to unpick the problem. So one of the first things I did was trying to develop a fair test for inbreeding, or inbreeding depression. This again it's not as easy as you might think, because there's so many, all the different breeders use slightly different versions, so the patterns of relatedness in boxer dogs are fantastically complicated. So what I did, 1, 2, oh yes, right. So for as many dogs as I have, and you would not believe the number of hours I have spent typing in pedigrees of dogs. I've got 7,500 individuals and both parents typed in. And what with the Van Ders and the VDs and the Vs and the V dot and the VD dot and the number of different ways people spell their different dog names, even though it's meant to be the same dog. Fortunately, I do some computer programming, which has helped, but I have now got a massive database, and I can type in many dogs, and my program will spew out a 5 or deeper pedigree, 5 generation or deeper pedigree. So here we have a typical pedigree, just in outline. We've got a dog that may have JKD, and then we've got its parents going back, as I say, at least 5 and often many more generations. And what we want to do is to ask whether or not there's evidence that the JKD in this dog is due to inbreeding depression. Well, of course, you could select your... Oh, it seems to be doing it automatically. Or are you preempting?
It's me, I'm watching.
Oh, right, okay, I was wondering. Right, so you could just simply compare the inbreeding coefficient of JKD dogs with a whole load of healthy controls. But that doesn't really work if you think about it, because what if most of your JKD dogs come from a small number of studs, which have been used extensively in a small, unusually inbred pedigree? Then they're going to be more inbred anyway, and the two factors are intertwined too deeply. So what I thought was the best control for how inbred this is relative, the best control for a dog is its parents, because its parents have come from the same breeding line and therefore will probably have experienced the same sort of patterns of breeding. So what I did was I compared the inbreeding coefficient of a JKD dog going back I've done it smaller to save having to do all these. Going back 4 generations, but then I take the parents and I take them back 4 generations, and I compare the difference between the inbreeding coefficient of the average of the parents with the JKD dog. And you get a small effect, which it's significant, it's only at 5%. I want to do it on even more pedigrees. So there is a small effect, but this does support what Bruce was saying about the possibility that this is a recessive factor where you need to get it from both parents, because you're more likely to get it from both parents if you're a bit more inbred than average. So there is a very small, but just about scientifically significant effect of inbreeding. Thank you. Right, this was taken off the web and was out of focus on the web, so sorry. But this is to illustrate, thank you, dogs, for having so many chromosomes. They've got 38 pairs of normal chromosomes plus the sex chromosomes. That's a lot of chromosomes if you want to start looking for a gene. I used a very broad brush approach to start with. I used a series of genetic markers. These are bits of the DNA which have lots of different types, so you can track them easily through pedigrees. Up to about 15 states, we call them alleles. And I used just a panel of, many people would look down their nose at this, just 70 markers. 70 markers means that I can put 2 or 3 on each of the larger chromosomes and going down and maybe just 1 on each of the smaller chromosomes. But at least I mark each of the chromosomes, which will then allow me to ask whether the gene is a bit more likely to occur on that chromosome in particular. And I did have one hit, and this illustrates it. So these are our, oh, the font has come out funny. Okay, well anyway, these are meant to be numbered, but you can see that there are a lot of them. These are the genetic states, and here are the frequencies of these states. So we've got state 1, 2, 3, 4, all these in healthy dogs, and you've got the same states in a whole bunch of affected dogs. And what you see is that they're very, very similar frequencies. So if it's common in the healthy dogs, it's common in the affected dogs, all the way through to this little group here, which all seem to be a lot commoner in the diseased dogs. And this is a significant effect. It's really quite significant. But there are a number of take-home messages, some of which are really good because they suggest that this is indicative of being close to the gene. And some of it is a bit of a pain. If you notice, there are an awful lot of these blue bars do not differ between diseased dogs and healthy dogs. So it's not like, right, I found it, this is 100% guaranteed genetic test. What I have is a case where there are a few of these states, a few of these alleles, which seem to be strongly indicative of being a carrier for the disease. For me, I was particularly convinced by this because, in fact, if you look at which ones seem to cause risk in different populations, they seem to be a bit different. So the US tends to have this one, whereas the British dogs seem to have this one, and the Scandinavians tend to have this one. Which is interesting, because that's exactly what you'd expect, because these markers, in this particular case, they tend to evolve by going to adjacent types. So probably these states are all related to each other if you go back a long, long way in time. So it's kind of what you'd expect if the gene was sitting next to an allele that had changed a little bit, but not too much. And so it sort of makes biological sense. So the positive news is this looks as if I've found a part of a chromosome which is sitting quite close to a gene. The negative bit is it's far from a magic test where you can test and you can say this dog will get the disease or this won't. A particularly interesting story was that Bruce told me about one of the top studs that was really used a lot and had produced many affected offspring. He said, this dog is homozygous. He carries 2 copies of the gene. Well, I could test this because I had 13 of his offspring in my sample set, and in fact all of them had allele risk 13. That was one of these alleles I'd identified as indicative of potentially related to the disease. So kind of, that's quite a coincidence. It's not a common allele, and yet Bruce had said this dog is homozygous. And in fact, I then found that a historical sample, we had a sample from the dog, and he was in fact homozygous for 13.
And was he healthy or healthy?
The complication is
Has
that he was
infections belly.
perfectly healthy, which seems to be this male-female divide, that it rather supports again the idea that Bruce was putting forward, which is that males seem a bit more tolerant of having a faulty gene, and maybe their kidneys can function on and they can push on into later life or even survive and not get kidney disease at all. But this just shows you that it's not a simple problem, and I think, I'm afraid the negative side of my talk is that really we're looking for something which probably doesn't have a perfect solution. It's not like you can do a test for Huntington's disease in humans and yet they know exactly what they're looking for, and if you have this particular characteristic, you will get the disease. So it's unlikely to be cut and dried, but I am optimistic that we will get closer. And in fact, this is what I've been doing recently. Here we are. It's at the end of chromosome 1, on one of the biggest chromosomes. This chromosome is about 165 million letters long in DNA terms. That's a lot of letters to look at. I've so far focused in on an area in which I started reading 3 million letters from each of my dogs to start looking to see whether or not I could find a gene in the surrounding region. This only produced results which were about similar to the one before. But I now believe, in fact, that because there was another marker that was further down here, which showed a medium strong signal, that probably the gene lies in this region here, slightly below the red line. So I'm now going off and I'm going to repeat it with another 6 million bases, but this time 6 million letters, but this time I've gone for a region that's a little bit further down, and I've got information back on a test run of 50 dogs, and it kind of looks promising, which is good. I hoped that I'd have the results for this talk, but you do 2 weeks of experiments, you then send stuff off to China, and 40 days later you get the results back. It's quite expensive, so you do a test first to see if it's working, and then you go back and do all the dogs, and then you find you've gone to the wrong area, so you do another test on the next lot, and then and each one is a 40-day lag. So unfortunately, I do get emails all the time saying, how's your research going? What are your results? And I have to say, they're still sort of brewing. But I hope to have my main analysis of this bit of the genome ready soon, and I'm optimistic that it will be a bit of a stronger result than I got before. And of course there is this, the sex bias, which I'm also starting to look into, and it's a good guide because actually it struck me that really I should be using all females for my main test because they're the ones with the disease, and that including males will kind of muddy the waters a bit because you get healthy males, as we know, who seem to carry the gene, but they're still healthy. So I think that's lots of thanks, particularly to people in the different countries who've been providing samples, and really in particular poor old Mathieu Brocheton, who was a French guy who was fantastically enthusiastic, and he unfortunately was killed in a car accident halfway through the study. But I mean, huge amounts of energy, and he managed to get all of his friends to send me samples, etc. And I have received some funding from the Council of Dog Breeds and the Kennel Club, which has allowed me to do this. Thank you.
Bill, thank you very much. Okay, gather your thoughts and your questions, hold on to them. We're going to have a coffee break, tea break, and we adjourn at 3 o'clock.